OEB's 20th birthday. And there's a lot of great things about OEB that we all know. But one thing about OEB that we students appreciate particularly is Darwin Fellows. And so for the first event of the day, we had 4 Darwin Fellows coming back to celebrate with us. And besides being a part of this festivity, they each is going to give us, tell us what has happened since their time in OEB. And I would love to introduce them to you guys, but there's a better person to do that than I am. And the person is our current Darwin Fellow, Sevan. So I'm just going to hand it over to Sevan and let her introduce her.
Well, thank you so much. It's great to see so many fabulous hats. And if you don't have anything on your hat, there are a few things that I can totally imagine you putting on your hat before this event is over. So it's my great pleasure to introduce our first former Darwin Fellow speaker of the day, Dan Ardia. He was here from 2004 to 2006, working with Yi-Fen, Felter. He works on the ecology and evolution of life history traits and ecological immunity in birds. And he will be talking to us today about...
I don't have a title.
You don't have a title. But
random things I've done since I left.
random things that he's done since he's left here. So please join me in welcoming.
I have a bit of a cold, I'm sorry. It's really a pleasure to be back. I haven't been on campus, I think, since 2007. Well, some things like some beautiful life science building north of here, which I guess the biology department isn't housed in. This building looks the same, but it's a pleasure to be back. So first I wanted to thank OEB for giving me the opportunity to be a Darwin Fellow. For me, it was a really important, formative experience to have that opportunity to do research, to teach, and also to be a mentor to the graduate students. And I've made lifelong friends who are part of the grad students when I was here, and it really was a great opportunity. The first thing I want to say is that as a postdoc, I did more writing. These are all the papers I wrote in my first year here as a Darwin Fellow, all data from my dissertation, and that was a tremendous opportunity. And I wanted to thank Yi-Fen for allowing me to work with him. So that's Yi-Fen there. We had a very productive time in the fields off Amherst College, and that really springboarded a lot of projects. So what I'd like to do is just give you an overview of the work I've been doing since I left here in 2006, I guess. So I'm going to kill that. That did not help, sorry. Okay, so funding at NSF is a bit dire right now, and I was lucky to get 2 NSF grants in my first couple years at F&M, and so I probably will never get another one the way funding rates go, but I'd like to talk to you about 2 projects I've been involved in. The first was a large 5-year grant through a program at NSF called the Partnerships for International Research and Education, PIRE, and it had 2 main goals. One was obviously to test a research question that I'll tell you about in a little bit, and the other was an education component, and so it involved bringing mostly recent college graduates to work in international research, to institution build in the countries in which we worked, to give students experience in working overseas. I think to just make better science citizens was the goal of the PIRE program. And so the project that we did was one across the Western Hemisphere looking at life history evolution in the genus Tachycynida, which includes the tree swallow. And so we had a very ambitious project that worked in some ways and not in others. And so each of these are different locations in which we have had field workers. And so we weren't actually allowed to spend money in the United States, but we still found ways to have collaborators out west, but we had a number of different sites in Canada. And then our main tropical sites, which I'll show you some data for in a second, are Belize and Venezuela, a site here in the dry forest of Peru, a number of sites in Argentina, including in Tierra del Fuego. And the argument that we made to the National Science Foundation was that to try to understand geographic variation in life histories, it's really important to control for evolutionary history, that looking at something like an ant shrunk and a robin, have very obvious differences in life histories, but it's hard to determine whether those differences are due to their very different evolutionary histories or due to the environment. And so we made the point that by studying a single genus that showed life history variation, that we could better control for evolutionary history and try to get a feel for the factors that drive life history variation. So we focused, at least initially, on clutch size, which we know are larger in temperate areas, both north and south. And this is an oversimplified diagram of the factors we think influence clutch size. And I'm just going to show you a bit of data today to reflect on that. But we had 4 general components we thought played a role. Seasonality, which of course has been a hypothesis proposed many times, and longer day lengths and longer breeding seasons. In the temperate zone, would allow birds to invest more in reproduction. And so we have a lot of data on active day and looking at whether or not birds are limited in time available for foraging in the tropics. We might call life history tactics, which is really a trade-off between survival and reproduction, and we're just getting the last season's worth of data over the 5 years in Belize and Venezuela this past year to be able to do some more sophisticated survival analyses. Environmental variation, which really is temperature, and then finally the density of predators, which we don't unfortunately have much data. So I'm just going to, so the project then morphed from not just clutch size, but it really became a project about integrating physiology and behavior across life history stages. So we were able to collect a lot of interesting information of really every stage of the bird's life histories, from the production of eggs, the number, the composition, of course the clutch size. In the work I did with Yi-Fen, I became quite interested in incubation behavior, and I'll show you that that's the main focus of my work right now. So we have a lot of data, and I'll show you some about birds' investment in incubation and how egg temperatures might vary, and then a whole variety of factors associated with the nesting period. So I'm just going to show you some tidbits of information now. And so one thing we find as a broad pattern is that parental investment varies in the way you would predict that in general tropical birds invest less per breeding attempt than do temperate birds. And one way in which we see that is in the amount of time birds spend on the nest incubating. And so here we're looking at 6 species, 3 tropical here, and a north temperate and a south temperate, looking at the percent time birds, females, spend in the nest during the daytime in the incubation period. And you can see, if you're not aware of this already, that small birds like swallows spend somewhere between 50 to 60% of the day in the temperate zone in the nest, not like ducks or chickens that are in there all the time. And the tropical birds spend a lot less time. And this raises a number of interesting questions about embryonic development. So here's a developing embryo that's getting only 50% incubation. So incubation periods are longer. Parents end up, I won't show you data on it, making more visits. So it has a whole suite of influences on parents in terms of rewarming the eggs, and on embryos in terms of suboptimal developmental conditions. We look at how much time parents spend feeding, and so these are data from automated perch counters that we place outside the nest. Every time the parents come in, they hit a little activity switch, and so we're able to measure how many visits they make over the entire nestling period. And we switch now to using radio frequency identification technology to get that. So we have a very large data set on parental investment. And so this just looks at the daily feeding events per nestling, so the number of chick feeds per day. And in general, we see a clustering with the 2 species here that are closer to the equator feeding less than temperate species. And this is on a per nestling basis. So there are more chicks here. So from a chick's perspective, they get fewer feeding events. And from a total feeding investment, this is much higher. So these would be 5 to 6 offspring, and these would be 2 to 4. So these parents are working much harder. And we have some great proposals pending now to look at some of the energetic consequences of that. Another interesting thing we looked at was the role of thermoregulatory performance as a way to assess cold temperatures and physiological development. So we developed a field method that we could do at all these sites where we would measure the ability of nestling birds to hold their body temperature against a cooling gradient. And so what we're looking at here are the final temperatures that the chicks are able to maintain against a 20-degree water bath in the field. And so for young nestlings, only H4, who have no thermoregulatory ability, you would expect them to cool down to close to 20, and in fact we see that. And the reason this site out here, the Chilean swallow from Tierra del Fuego, is so low is it's colder than 20 degrees in the field, so you can just, it's kind of an outlier that's confounded by the cold environmental conditions. But at H4, there's no difference, as we would have predicted. Regardless of the species, everyone thermoregulates very poorly. But if we look at day 10 nestlings, and so here would be, for example, Tecustinetta bicolor, the tree swallow, here's the violet green, and latitude similar to ours. These are birds that are maintaining body temperatures against the cooling gradient, not quite at parental levels of 39 or 40 degrees, but they're still able to keep themselves warm against the cooling gradient. But here, the tropical species have still limited performance. Now, you could argue in two different directions, and we need to look at this in more detail, is that this represents just slower physiological development, but you could also argue in the tropics it would not make sense to build a machinery, the cellular machinery, which would be costly to thermoregulate if it's actually not that good. So I just wanted to thank my collaborators. It's mostly a group at Cornell I collaborate with, but we have a variety of collaborators and funding sources from all across Latin America and Canada. So that's one big project that I've been involved in since I left, and we're sitting on a lot of data, and I'm on sabbatical next year, so I really look forward to making a slide like that first one I showed you, filled with titles from lots of papers, so I hope I get time away from my teaching schedule to write. Also fortunate to get a grant from the National Science Foundation for a research coordination network. Some of you may be familiar with these RCNs. It's an effort to bring together people of a variety of different fields to kind of synthesize and move a field forward. So with my colleague Marty Martin at University of South Florida and Dana Hawley at Virginia Tech, we created a research coordination network in ecoimmunology. We're in the last year of that grant right now. It's funded workshops in Scotland and Germany, and less exciting places like Tampa and Ann Arbor. Our final workshop is in a couple weeks in Woods Hole, actually. We've funded graduate student exchanges to go to labs to learn techniques, and funded a variety of different efforts. And it's been a great opportunity, I think, to meet different people and build collaborations. So we're fortunate to have done that work. Talk a little bit about work I do more close to home in Lancaster, where I live. Jeff Potos' alma mater at Franklin and Marshall College. So I'm going to focus on incubation and egg temperature as a way to assess parental investment. So one thing that's clear is that people that look at incubation underestimate the value of egg temperature. So you can measure how much time birds spend in the nest, but it doesn't really tell you the true conditions that an embryo experiences, because parents could modulate temperature in different ways. And so this just to show you that this is a trait that varies. So these are females incubating eggs in the same place in the same season at the same egg age, and incubation temperature varies widely. Some birds maintain warmer temperatures, others lower. So this is so we've done quite a bit of field respirometry work where we're able to hold eggs at different temperatures and look at their thermal sensitivity to see how the embryos respond to varying temperature. And so this is just a schematic of the research outline. So here I'd be interested in this case, a single trait egg temperature, looking at a variety of different factors. And so you could imagine, of course, the characteristics of the female would play a role, the eggs themselves in terms of their hormone, their own metabolism, their sensitivity. Of course, the environment going to influence the behavior of the parent, which is going to influence the egg temperature, and then have a whole suite of cascading effects on physiological development. And so my site in Lancaster has been shut down, though, because of snakes. A lot more snakes in Pennsylvania than there are here in Massachusetts. For a couple seasons, we were pulling big 6-foot snakes out of our nest. That's a king snake there, but often we get 6-foot black rat snakes in our nest. They would learn a trap line and stuff. And here's a snake full of 5 house wren babies. So I've shifted to do some of this work in the lab, and so what I'm just going to show you a little tidbit of data here on the role of developmental temperatures, in this case on a physiological metric, a very coarse measure of innate immunity, the ability to kill bacteria. And so here are 2 species, one altricial zebrafinch, one precocial, the bobwhite quail, raised at 2 different temperatures in an incubator, 36 and 38. And their performance of innate immunity as embryos and as juveniles. And so one thing you can see is that embryonic birds have no ability to kill bacteria. So the thing to keep in mind here is the manipulation is occurring when they're embryos. And so I'm arguing this is a form of perinatal programming, that it's not like they're developing it during their embryonic stage, but rather environmental conditions during the embryonic stage is somehow acclimating them when they get older in terms of which aspect of physiology to potentially invest in work, some sort of a constraint. So we found then that as they hatch, that those that come from the colder temperature conditions in both the altricial finch and the precocial bobwhite quail have lower performance. So we're trying to follow that up a little bit. So in another proposal I have pending is to look at the effect of early life experience on late life phenotypes, and in particular the early life experience is temperature, changing mean temperature, changing temperature variance, to see how that affects a variety of different metrics of performance and what the mechanism is. So I built a team I'm handling the metabolic rate and the thermoregulation. I have recruited an immunologist who's going to look at a variety of different factors. People interested in oxidative stress and stress physiology, so we'll see whether it's transformative enough for the National Science Foundation. Very briefly, I'm going to tell you about some more fun projects. Not that those other ones aren't fun, but they're more serious, knowledge-advancing sorts of things. And these are just things I like to do with my students. I like to misnet birds, and so we've set up an array of feeders, looking at wintering birds, putting RFID pit tags on them, and putting little circuit boards out so we can see what they do, and we can generate data that looks like this. So this is just a handful of chickadees from last winter. They have an array here of 6 feeders they can choose from. And so we're able to do kind of a basic network analysis, and we're looking for money now to kind of step up the database aspects of it. But you can see birds have different preferred feeders. This one loved feeder 3, this one loved feeder 6. And we can look at its transition probabilities between feeders, how they spend their time. And now we're trying to map on some measures of coping styles or behavioral syndromes to look at physiological and genetic factors that predict which birds move more than others, those kinds of things. So. Also started looking at red foxes, which are very common in urban areas in Lancaster. So you have a lot of coyotes around here, which means that you have a lot fewer red foxes than we do. We're at the northern, right just below the southern edge of coyotes in Pennsylvania, so we still have a lot of red foxes in our city. So we've been putting out game cameras on an urban to rural gradient to try to look at where the foxes are, and so we get normal looking pictures like this, and this cool ghostly picture of a fox checking out our camera. We've done a bunch of different things. Just for my last little tidbit here, I want to tell you about a study my honor student did last year looking at the role of anthropogenic food sources on foxes in urban areas. So here's our sites. The blue areas are places where we captured foxes, and these are foxes that were trapped by a local trapper who gave us the fur. And so what she was interested in is whether or not, I'm sorry, the axes got screwed up, is whether or not stable isotope ratios in the fur of the foxes would indicate levels of anthropogenic food. So humans eat a lot of high fructose corn syrup, which means that our garbage has a lot of high fructose corn syrup in it. Corn has a different stable isotope signature than presumably wild foods, although we know they're eating things that eat corn, so I realize that's a problem. And so in general, we find that, that there's good predictors of foxes that live in places that have a lot of developed land, in particular industrial areas. Foxes love because they're busy during the day, but then no one's there at night, so they can come and do what they want. Predicts the amount of garbage, essentially, they eat. So we're interested in how they're adapting to the local landscape. So. And that's it. So thank you for listening.